Hello everybody, my name's Dr. Mark Temple. I'm based on the Campbelltown campus, University of Western Sydney, and I'm going to be presenting to you a series of 4 lectures for the unit Molecular Biology. These lectures are taken from the textbook, and as you know, that's Molecular Biology by Cox et al. And this lecture is based primarily on chapter 18 about protein synthesis. So, as a broad overview of the process of protein synthesis, it can be broken up into 3 stages, and these are the stages of initiation, elongation, and termination. So effectively, initiation is the bringing together of the cellular machinery required to begin the process of protein synthesis. Once this has been achieved, we then go through the process of elongation, where amino acids are joined together sequentially to form a string of amino acid residues, and this becomes the protein. And then once the protein's finished, we then terminate the synthesis of the protein, and then it's released into the cell to perform its function. In addition to these 3 processes, we have to consider that the amino acids need to be activated so that they can be incorporated into the protein. So we have a separate process of amino acid activation. So AA here refers to amino acid. So we have amino acid activation, and then these amino acids feed into the process of initiation and there's sequential rounds of elongation. And once the protein synthesis is terminated, the protein then needs to be folded so that it can take on its correct structure so that it can perform its cellular process. So the big question is, how do we go from a messenger RNA sequence to a protein sequence? So the messenger RNA is a string of nucleotides, and as you probably know, 3 nucleotides form a codon, and each codon specifies for a particular amino acid. So for each codon within the messenger RNA, this codes for a particular amino acid, and there are 20 amino acids clearly. So something exists that can read a messenger RNA nucleotide sequence and that can specify a particular amino acid. So there's some adapter, and again, as you probably know, this adapter is a transfer RNA. And so if you think about it logically, for each codon, there's a separate adapter or transfer RNA which can bring in one of 20 individual amino acids. And then these are strung together by peptide bonds, so we have to have a process of peptide bond formation to string together the amino acids into a protein. So back in the 1950s, people postulated that this adapter existed, and this adapter was the link between a messenger RNA nucleotide sequence and a particular amino acid. And as we know, this adapter is a transfer RNA. So we have the messenger RNA, which contains the DNA sequence, and then we have the transfer RNA, which has an anticodon so that each codon can be read, and then each transfer RNA is attached to an amino acid, and it brings the amino acid into the process of either initiation or elongation. So at some point, the amino acid has to be attached to the adapter, and there's an enzyme in the cytosol that performs this task to link the transfer RNA to the amino acid. This enzyme is an amino acyl tRNA synthetase. Now, there's not just one enzyme. Remember, there are 20 amino acids, so there are at least 20 different tRNA synthetases to make these building blocks here, these transfer RNAs attached to their amino acid residues. So we have a pool of tRNA synthetases that are able to attach individual amino acids to specific transfer RNAs so that they can then be used in the process of initiation and elongation. Now, here's a table from the textbook which looks rather overwhelming at first, but if you break it down, it's actually a great summary, and there's a lot of information here, so it's like a good crib sheet to look at the process. So here we have a process of initiation, elongation, and termination that we'll be talking about. And also we have the activation of amino acids, and we have the process of folding and making the tertiary structure of the protein. So here's a list of what we'll be talking about in the lecture. A lot of this lecture deals with what's known in E. coli, and this is a fundamentally important conserved process. So a lot of what we understand from E. coli clearly translates to what we know and understand in eukaryotes. So this table looks overwhelming, but it's a good summary of all the key elements that we'll be looking at during the lecture. So I would come back to this once you've studied the material, and it'll make a lot more sense to you. So the first thing we need to consider when looking at translation and protein synthesis is the big molecular machine that performs this in the cell. And the ribosome is the big molecular structure that we're going to be looking at where protein synthesis occurs. So the ribosome is a large structure made of many proteins and RNA, and it facilitates protein synthesis, and it brings together the messenger RNA containing the codons. It brings together the transfer RNAs, which are attached to the charged amino acids, and then within the ribosome it catalyzes the peptide bond formation between the amino acids to form the polypeptide and protein. So here's a cartoon structure of a ribosome. I based these cartoons on the images in the textbook, but rather than use the textbook images, as you'll see, I've created these cartoons so I can try and animate the process for you to explain to you what's actually happening. And I strongly encourage you, as always, to read the textbook covering the information that I'm presenting. Don't rely solely on my presentation. Clearly, you must read the textbook. So here I'm showing you a cartoon of a ribosome. So we have the large 50S subunit of a ribosome, and then the smaller 30S subunit of a ribosome. Now between these two large structures, the ribosome forms three pockets, if you like, three sort of active sites, and referred to as A, P, and E. And I'll describe to you what these sites are. But you'll notice these sites span the two ribosomes. So the A site is made up of part of the 30S ribosome and part of the 50S ribosome, as are the other two pockets. They span both ribosomes. And if you also note here that in the smaller 30S subunit of the ribosome, it's shown binding to the messenger RNA. Now, within these A, P, and E sites of the ribosome, of the formed ribosome, there are two important catalytic centers, or important functional centers. In the 30S subunit, there is what we call a decoding center. So this is where the codon-anticodon interaction occurs between the messenger RNA and the transfer RNA. So the codon of the messenger RNA base pairs with the anticodon of the transfer RNA to establish information flow from the messenger RNA to the adapter. During the process, another process I'll describe to you amino acids are specifically attached to an adapter so that when this base pairing here occurs, it brings in the right amino acid. And then in this subunit of the ribosome, in the large 50S subunit of the ribosome, there's a peptidal transferase center. Effectively, this subunit catalyzes the peptide bond formation. And we'll look at all of this in a bit more detail. Again, here's a table from the textbook, and this is just summarizing the E. coli ribosome. So here we have the two subunits of the ribosome. We have the 30S and the 50S subunits of the ribosome. So here's the 30S subunit and the 50S subunit. The term S is you don't need to know, but it's a sedimentation coefficient. So when they first identified ribosomes, they would have spun them down in some sort of centrifugation process, and they would have separated out at different weights, different positions, and then they refer to them as having a sedimentation coefficient. But effectively, we're just referring to 30S as the small subunit and the 50S as the large subunit. So for these two subunits, this is a brief breakdown of what each subunit contains. So the small 30S subunit is made up of 21 individual proteins, and it's made up of a large ribosomal RNA, so an rRNA. And the 50S subunit is made up of 33 different proteins, and there's 36 proteins in total, so some of them occur more than once. And then within this larger 50S subunit, there's also two large RNAs. So we have the 5S RNA and the 23S RNA. So effectively, we have three RNA species in this process. We have the messenger RNA, we have the transfer RNA, and we have the ribosomal RNA. So when people first started to look at the process of protein synthesis, it was probably assumed that protein enzymes would be involved in catalyzing peptide bond formation, that proteins would be involved in the process of protein synthesis. It turns out that they were wrong. Effectively, the fundamental process of protein synthesis is catalyzed by RNA, not protein. And this was a major surprise, and it's kind of fascinating that we have not only proteins with enzyme activity, but we also have RNA with enzyme activity. So the ribosome it's a mixture of proteins and RNA, but it's the RNA within the ribosome that's really driving the process of protein synthesis. So given the importance of ribosomal RNA in the process of translation, it's not surprising that these RNAs have been conserved across different species. So here's this cartoon here represents the folding of a single-stranded RNA into a structure, and if you look at the RNA from the small subunit of the ribosome in bacteria, archaea, and eukaryotes, you can see a lot of this structure has been conserved across these very different species. And that's important because all of these species obviously have protein translation as part of their fundamental processes, and clearly the RNAs have been conserved. These are also interesting, and you've probably not seen these sort of structures before. Effectively, all RNAs are single-stranded, but within that single-stranded structure, there's regions of base pairing to form these fairly complex three-dimensional structures. And this is just a two-dimensional representation of these ribosomal RNAs. And these were determined before X-ray crystallography 3D structures were known. So these are drawn based on the complementary base pairing of the rRNA. So the rRNA is responsible for all of the major functions of the ribosome, and these include the transfer RNA binding to the ribosome, peptide bond formation, and the coming together of the two subunits is all determined by the RNA that's within the ribosome. It's not the protein that's driving these processes. So the ribosome is clearly a very large, complex structure. I showed you those cartoons of the RNA based on the base pairing ideas. Now, since those early investigations, people have determined the X-ray crystallography structure of ribosomes, and here's one here from the E. coli showing the 30S subunit as a structure with lots of proteins and this big RNA here. Okay, and this is the RNA I was showing you a couple of slides ago as a cartoon with base pairing. This is the 3D structure of the RNA. And likewise, the 50S subunit of the ribosome also contains RNA and proteins. And if you look at this structure here, there's clearly a cleft here where you can get movement of transfer RNAs in and out of the subunit. There's also regions where the messenger RNA can thread through the small subunit, and there's also a region where the protein can extend out of the subunit. And again, these things are conserved across species. So this is the ribosome structure from eukaryotes. Again, it's a slightly larger structure with more proteins, but primarily it's highly conserved. I mean, clearly there are differences, but it's highly conserved, and it performs the same function with a cleft and a region where the messenger RNA can thread through, and then another region where the protein can emerge, and a region where the transfer RNAs can move in and out of the ribosome. Early studies of protein synthesis, they used electron microscopy to look at the cell, and here you can see in this faint blue is a string of DNA. Okay, and then from this string of DNA we have transcription making the RNA. So the RNA is being peeled off from the DNA. Now, as that's occurring, the ribosomes are attaching to the messenger RNA to undergo protein synthesis. So we have these overlapping processes occurring here. And again, this is taken from a figure in the textbook, and I've tried to animate it to sort of explain what's happening here. But here's our double-stranded DNA. Here's our polymerase, which is copying the gene into a messenger RNA. So here's our single-stranded messenger RNA. And then as this process of transcription is occurring, then on the messenger RNA, the process of translation, protein synthesis, is occurring. So here we see the assembly of the ribosome, and then these little dots here represent the amino acids being strung together. And as the polymerase moves along transcribing the DNA, the messenger RNA grows and more ribosomes attach to the messenger RNA, and likewise these processes are coupled so that as the messenger RNA grows, more ribosomes attach and produce the protein. So the ribosome is described as a ribozyme, indicating it's an enzyme. It means that the catalytic function is driven by RNA. And two pieces of evidence that really nailed this point was that the ribosome can be inactivated by making base pair changes to the rRNA, which indicates how important the ribosomal RNA is to the process. And also, if you can break apart ribosomes and deprotonate them, but the peptidal transferase activity, in other words, making the peptide bond, that activity is retained within the RNA in the absence of proteins. So it's the rRNA that's making these peptide bonds. So looking at the ribosome in a bit more detail, there are three sites that are important for function. We have the A site, P site, and E site. So as we'll see in some of the slides coming up, the incoming amino acids attached to the transfer RNA enter at the A site. The growing peptide chain is located in the P site, and the E site is where the transfer RNA exits after it's donated its amino acid to the polypeptide. So effectively each transfer RNA bringing the amino acid, it enters the A site, it then moves to the P site after peptide bond formation, and then it exits the ribosome via the E site. So it's this big complex molecular machine where a transfer RNA comes in, donates its amino acid, and then leaves. And the next one comes in, donates its amino acid, and then leaves. And this process is run by the ribosome. So here's a diagram showing the A site, P site, and E site of the ribosome. Recall that the 50S component of the ribosome has the peptide bond formation activity, and effectively during the process of elongation we have these sites occupied. So each amino acid comes in the A site, it then moves into the P site, and then it exits from the E site, and the process of peptide bond formation occurs as these transfer RNAs are positioned within the ribosome. Here's the 30S component of the ribosome, and here's the 50, so the small and large subunits with the proteins and RNAs color-coded. And shown here are three transfer RNAs in green. Okay, so here's the A site, here's the P site, and here's the E site. So the transfer RNA enters here. Now, again in red shown here is the messenger RNA embedded within or threaded through the small subunit. The base pairing between the transfer RNA and the messenger RNA occurs here, and then once the right transfer RNAs are brought into place based on the messenger RNA sequence, the process, these purple things here are the amino acids. These amino acids are then strung together by the catalytic activity of this RNA. So this big RNA here is performing the catalytic activity of making a peptide bond between these two amino acids. And this is just showing the arrangement of three transfer RNAs within the ribosome, but they've taken away the ribosome so you can see it in a bit more detail. So if you look here in this previous slide, buried in here is a messenger RNA that you can't really see, and buried in here are three transfer RNAs which are quite difficult to make out. But if you isolate those, then here's the messenger RNA and here's the transfer RNAs. You can see here from another angle that we have the base pairing between the codon and the anticodon here, which is transferring the genetic information from the messenger RNA specifically to a unique transfer RNA. And that transfer RNA is attached to a particular amino acid, and within the ribosome structure, these amino acids can then be joined together in a peptide bond to form a polypeptide. So if you look at this little cartoon here, it's the same kind of information. Here we have the messenger RNA threaded into the small subunit. We have the base pairing between the codon, anticodon of the messenger RNA, transfer RNA, and here within the large subunit we have the catalytic activity, which is that RNA which will make the peptide bond between the amino acids. And again here the anticodon loop and the codon of the messenger RNA come into contact here, and that determines the information transfer from the messenger RNA to the right amino acid. So here we have a cartoon from the textbook which represents the process of translation. So we have a stage here which is the initiation of translation, in other words the coming together of the messenger RNA and the ribosome and the starter transfer RNA. And then once this is assembled for initiation, we then go into the process of elongation, where different amino acids enter and are strung together. And then once elongation's finished, we then have a termination, and then these components can be reused to make more proteins. So we'll be looking at these processes in a bit more detail. So here's another 3D crystal structure of a ribosome, and this one's interesting because it shows the position of the messenger RNA within the 30S subunit, within the small subunit. And what you can see here is that the messenger RNA is unfolded and strung out into a sequence which can be easily read. Okay, and it forms this kink here. Now, this unfolded messenger RNA can then bind to the anticodon. And then what's also shown here is, apart from the cleft or the hole where this can thread through, is another tunnel within the ribosome where the synthesized protein can exit. So we have the transfer RNAs coming in and out of the ribosome, we have the messenger RNA threading through the small subunit, and we have the peptide exiting through the large subunit. And as the proteins are made, clearly there's no folding occurring within the ribosome because it's strung out. But as it exits the ribosome, it's then able to begin the process of folding. So to finish off this section of the presentation, I'd just like to refer to some of the summary points from the textbook. And these are more of a dot point presentation rather than sort of a graphic presentation. So the protein synthesis occurs on ribosomes, and the ribosome is a large complex of protein and RNA. The ribosome is made up of two subunits, and in bacteria we refer to these as a 30S and a 50S subunit. And these two subunits come together to form a subunit with a sedimentation coefficient of 70. Okay, so it's a larger sedimentation coefficient, which is the combination of these two smaller subunits. And in eukaryotes, the basic structure of the ribosome is highly conserved, but the overall structure of the ribosome it's larger and it's got more proteins, but performs the same function, and a lot of the critical regions are highly conserved. To summarize the process of protein synthesis, then effectively it's regulated by the binding of the small subunit to the messenger RNA before translation begins. So we assemble the machinery before we start the machinery, effectively. And once the ribosome is fully assembled, it then moves along the messenger RNA, and as the ribosome moves along the messenger RNA, it matches individual transfer RNAs to the codon of the messenger RNA. And on a single messenger RNA, you can have multiple ribosomes performing this process. Like I said, this process is highly conserved, so in all organisms, the rRNA of the large ribosomal subunit performs the catalytic activity of peptide bond formation, and the small subunit of the ribosome reads the genetic code and brings together the right transfer RNA, which brings the right amino acid in for peptide bond formation. In all cases, the amino acyl transfer RNAs, they join the ribosome at the A site, and then peptide bond formation occurs, and then the tRNA, which now doesn't have an amino acid attach exits through the E site. And again, in all cases, the messenger RNA is able to enter through channels on the subunit, and the growing peptide is able to exit from channels in the large subunit. When the ribosome binds to the messenger RNA, it unfolds the messenger RNA, allowing interaction between the transfer RNAs and the codons. And as the peptide leaves the ribosome, it's then able to begin the process of folding into its correct structure. The next section of the textbook concerns the activation of amino acids for protein synthesis. So this is the process that occurs before initiation. So the activation of amino acids doesn't occur at the ribosome. This occurs in the cytosol, and then once these activated amino acids are attached to the transfer RNA, these transfer RNAs move to the ribosome during initiation and elongation. We talked earlier about the fact that amino acids need to be linked to specific transfer RNAs, and the amino acids need to be activated for the process of protein synthesis. So this activation is referred to as charging of the transfer RNA. That's not referring to a positive or a negative charge, but it refers to charging of the transfer RNA, meaning that the transfer RNA contains an amino acid ready for protein synthesis. So the charging of the transfer RNA requires the formation of an acyl linkage between the amino acid and the transfer RNA. And this occurs at the carboxyl group of the amino acid and the 2 or 3' OH of the sugar of the transfer RNA. So if you recall from your what you know about nucleic acids, that the backbone consists of sugar phosphate backbone with bases attached. At the end of the transfer RNA, there's a sugar with a free 2' and 3' hydroxy. Now, at the end of the transfer RNA, at this sugar will be attached to an amino acid at the carboxy group of the amino acid. So I showed you earlier a slide like this indicating that an amino acyl tRNA synthetase protein was responsible for the linkage from an amino acid to a transfer RNA. And I also pointed out that there are 20 of these enzymes because there are 20 different amino acids. So there were at least 20 of these enzymes to make these transfer RNAs. Now, the amino acyl tRNA synthetase activation of amino acids is a two-step process. So it's a two-step reaction. The first step here is the adenylation of the amino acid. So the amino acid is bound to a nucleotide, and the ATP is attached as AMP to the nucleotide. The second step of the reaction is the transfer of this adenylated amino acid onto the transfer RNA to give the transfer RNA with the charged amino acid. So this synthetase here performs two steps, which is the adenylation of the amino acid and then the transfer and charging of that amino acid onto a transfer RNA. So here's a figure from the textbook that shows that first step of the activation of amino acids, which is the adenylation step. So here's our amino acid. Here's the carboxy group. Here's the R group. Now this carboxy group is attached to an adenine nucleotide, so ATP, and two high energy phosphate bonds are cleaved here, and the phosphate group here is attached to the carboxy group to form this amino acyl activated amino acid. So here's the amino acid now attached to this adenyl group. Now that's the first step in the activation of amino acids. The second step is the attachment of this to a transfer RNA, which is shown here. So the second step takes this activated amino acid and it's charged and it's added to a transfer RNA. So there are two different paths it can take, but they're both very similar. We have a transfer RNA, and the last nucleotide of that transfer RNA happens to be an adenine. This adenine of the transfer RNA is then attached to the carboxy group of this amino acid. So this intermediate step here is then used to transfer the amino acid to the transfer RNA at either the 2' position or the 3' position. Okay? And there's two classes of synthetases that perform slightly variants on this reaction, but the endpoint's the same because effectively we have a trans-esterification, are both converted into one form, which is the amino acid attached to the 3' hydroxy of the terminal adenine residue of the transfer RNA. So this sort of little green blob here is the book's representation of a transfer RNA. This here is the last nucleotide of the transfer RNA, and it is now attached to an amino acid. So this is the adapter molecule, which has a anticodon that can read the codon of the messenger RNA attached to an amino acid. Now, the attachment of an amino acid to a transfer RNA has been referred to as the second genetic code. Okay, so it's really important that the right amino acid be attached to the right transfer RNA before it goes to the ribosome. So to highlight the importance of the synthetase interaction with the transfer RNA, we have these crystal structures here. Now there are two types of synthetases. It doesn't really matter for what we're talking about here, but in both cases you can see there's a very large region of interaction between the transfer RNA and the synthetase. And this interaction here is referred to as the second genetic code. And this very specific interaction here is vital to attach the right amino acid to the right transfer RNA. And again here we have the transfer RNA and the amino acid, and again there's a lot of interaction between the enzyme and the transfer RNA to establish that the right amino acid is attached. So here we have a clover leaf representation of a transfer RNA, and shown here is a color coding of the bases to indicate how similar they are between different transfer RNAs. So the 3' terminal of all transfer RNAs contains CCA. So C, C, and then the A. Now the terminal A is where the amino acid is attached, as shown in those previous slides, where the amino acid activation occurs and is attached to the terminal adenine. Okay, and that sequence is conserved across all transfer RNAs. And don't forget, the cell's got more than 20 of these transfer RNAs. Unique to each transfer RNA is the bases here. And this is because this forms the anticodon that interacts with the messenger RNA codon. And amongst the remainder of the transfer RNA are specific bases for each transfer RNA that determine that interaction with the synthetase. So that second genetic code is determined by specific bases of the transfer RNA to the specific synthetase. And just as a reminder, the terminal residue here is an adenosine where the amino acid is attached. Now, this is exactly the same information as here. This is a 2D structure based on hydrogen bonding before X-ray crystallography data was known. Here's the X-ray crystallography data of a transfer RNA. And again, the cloverleaf structure now looks a bit different, but here's the anticodon region here. Here's the CCA where the amino acid is attached. And those other arms of the cloverleaf structure form this 3D structure here. And then this shaped transfer RNA is what's shown in this figure here, which is that sort of angled structure here is actually the transfer RNA as a 3D structure shown here. Now, within the synthetase, there's a bit of error checking that occurs. So for amino acids that are similar, such as isoleucine and valine, are quite similar structure, the valine tRNA synthetase has to be able to distinguish between similar structural amino acids. So in this case, the valine synthetase has a pocket that binds the amino acid that is small, that cannot fit the isoleucine. So the small pocket can't accommodate an isoleucine, it can accommodate a valine, and therefore it can then attach that amino acid to the transfer RNA. The other situation is, how do you distinguish between a smaller and a larger? Because what's happening here is that the larger one can't attach, but within the enzyme for the larger one, wouldn't the smaller one be able to attach? So if this is the other synthetase here, the isoleucine tRNA synthetase, the pocket's large enough to fit the incorrect amino acid, but it has a proofreading pocket that's smaller, and if the incorrect amino acid fits in the smaller pocket, then it's removed. So effectively, the isoleucine tRNA synthetase is able to proofread the fact that the wrong amino acid is incorporated by having this smaller pocket for proofreading. So even though the synthetase is able to distinguish quite well, the different amino acids, there's still a high rate of error incorporation. It's about 1 in 10 to the 4, which is much higher than in DNA synthesis, is effectively what we're comparing it to. Now, it's not such a problem for the cell if a wrong amino acid is incorporated into a protein, because from one messenger RNA, many proteins are made, and therefore, if one out of 10,000 proteins has the wrong amino acid in it, it's of no real consequence to the cell. And also, proteins are broken down and not passed on during cell division. So these mutations in proteins are transient rather than genetically inherited. So it's of little consequence. So again, to look at the summary points from the textbook, the amino acyl tRNA synthetases are the enzymes that covalently link amino acids to transfer RNAs, and this occurs in the cytosol away from the ribosome. The acylation, it charges the amino acid so that it's got a high energy bond that can then, the breaking of this bond during peptide bond formation drives the process. And lastly, within the structure of the transfer RNA is an anticodon that's responsible for the binding to the messenger RNA, but there are other regions within the transfer RNA that determine the specific interaction with the synthetase. So the RNA has two functions. It specifies the exact amino acid through interaction with the synthetase, and the RNA also has an anticodon that specifies which codon it will recognize in the messenger RNA. So let's move on now to the initiation of protein synthesis that occurs at the ribosome. So effectively, the initiation is the assembly of the molecular machinery required to begin protein synthesis. So the assembly of the ribosome around the messenger RNA is shown in this cartoon here. So here's the messenger RNA, and here's the start codon, the AUG start codon. And the first thing that happens is the small subunit binds to and threads the messenger RNA through that channel we were talking about. And the start codon is positioned at the APE site. So at the P site, the start codon is positioned. So once this structure is assembled, the initiator transfer RNA is able to bind. So the initiator amino acyl tRNA is bound to the start codon. And then around this small subunit, the large subunit can then bind. Now, the amino acid incorporation that begins the process of protein synthesis is a derivatized methionine. So it's N-formyl methionine, as indicated here by F-met. So the first amino acid residue in bacteria that begins the process of protein synthesis is a special version of methionine. So it's probably apparent to you that AUG also codes for methionine, not just the start of initiation. So the cell has to have a way here to distinguish the methionine codon that is actually the initiator codon. Now there's an upstream sequence here, and the first AUG from this upstream sequence is what determines the assembly here of the small subunit. Now what we have, there's a sequence here in the messenger RNA that's referred to as the Shine-Delgado sequence, okay, the AGGA-GGU. This is the Shine-Delgado sequence upstream of the first AUG, and that distinguishes this AUG as an initiator codon rather than a plain old codon for methionine. This Shine-Delgado sequence is complementary to the sequence of the rRNA that's a part of the ribosome. So this specific base pairing here between these two sequences positions the messenger RNA so that the AUG site is then located in the P site here for the initiation. So that's how the cell is able to distinguish the first AUG to begin initiation. So here we have the Shine-Delgado sequence of the messenger RNA showing that it's base pairing to a region of the 16S RNA sequence. So this is just showing here the Shine-Delgado sequence in the messenger RNA. It's a conserved region that occurs in the majority of messenger RNAs, and the first AUG downstream from the Shine-Delgado sequence indicates that this AUG sequence will be the initiator codon. And this is just showing here that the methionine attached to the transfer RNA is modified into this N-formyl methionine, which then serves as the initiator. And in eukaryotes, there are two transfer RNAs for methionine, one which recognizes the initiation codon, and another that brings in methionine as part of the amino acid sequence of the polypeptide. So here I have a little animation which is again looking at the process of initiation of protein synthesis in bacteria, again with a little more detail. So what's shown in this animation is the inclusion of these initiation factors in the process. Now, so recall that the small ribosomal unit binds to the messenger RNA, and with the help of the Shine-Delgado sequence, it positions itself at the first AUG. Whilst this process is occurring, there are also these initiation factors, shown here in blue, IF1 and IF3, initiation factors 1 and 3. These bind to these A and E sites on the ribosome, and it prevents the formation of the full ribosome before the assembly is complete, if you like. So these factors bind and stop the binding of the large subunit. So shown here is the initiator transfer RNA bound to IF2, which is a GTP binding protein. In this case, it's bound to GTP. And both the initiator transfer RNA and the IF2 join the complex. And then there's a hydrolysis of the GTP, which is an energy-driven, an energy-dependent step, which then signals to the cell that it's ready for the larger ribosome to come in, and then the large subunit comes in and forms this complex. I'm not going to go into any detail of this in eukaryotes, but this same process is fundamentally conserved in eukaryotes, except that there are additional proteins that have become involved in the process. So in eukaryotes, the process is generally similar to what's observed in bacteria cells. There are additional initiation factors required for initiation in eukaryotes, but effectively the initiation factors that I showed you, 1 and 3 in bacteria, are conserved in eukaryotes. In bacteria, we have the Shine-Delgado sequence involved in the process. In eukaryotes, we don't have the Shine-Delgado sequence, but we have what's called scanning, so that the messenger, the ribosome scans along the messenger RNA in the 5' 3' direction until it finds the first AUG sequence. So just looking at some of the summary points from the textbook. In bacteria, all proteins begin the initiation of protein synthesis with a particular amino acyl transfer RNA. And this brings in a derivative of a methionine residue as the first residue in all proteins. In eukaryotes, there's a normal methionine is brought, but there's a special transfer RNA for initiation, and then there's another transfer RNA for normal methionines in the protein sequence. So in bacteria, there are 3 initiation factors involved in the formation of the initiation complex. IF1 and 3, they initially bind to the small subunit of the ribosome and prevent the binding of the larger subunit. IF2 is also involved in the initiation process. IF2, as I showed you, is bound to the transfer RNA for the initiator methionine, and this IF2 helps recruit this transfer RNA to the P site of the ribosome. And hydrolysis of the GTP then allows the dissociation of these initiation factors and allows the assembly of the large ribosome subunit to the complex. So once we have the activation of amino acids and their attachment to transfer RNAs, and we have the formation of the initiator complex at the ribosome, we can then begin the process of elongation, which is the addition of amino acid residues to the growing polypeptide chain. So effectively, the new nascent polypeptide is lengthened by covalent attachment of successive amino acid residues. Each amino acid is carried to the ribosome by its transfer RNA, and it's the transfer RNA base pairing to the codon that determines which amino acid residue will be added. So here's an animated sort of cartoon of the process of elongation. So the first thing we're showing here is the binding. So here we have the initiator complex with the first residue attached at the start codon. We have the A site that's free and the E site that's free. Here's the next transfer RNA, amino acid, which the anticodon matches the codon here. So there's a process of some helper proteins such as TU-GTP, which bind to the transfer RNA, and there's matching here of the anticodon-codon, and the amino acyl transfer RNA binds to the ribosome. So the function of this TU-GTP is to pause so that the peptide bond formation isn't instant, okay? And during this slight pause, it allows proofreading to make sure that the right codon is matched to the anticodon here. Once this is satisfied and the GTP is hydrolyzed, it then dissociates these helper proteins, and there's a rearrangement here of the transfer RNA to bring the two residues in close proximity, and then the process of peptide bond formation occurs, because here in the large ribosome is the peptidal catalytic center, that RNA we were talking about that has the enzyme activity, which will form the peptide bond. These helper proteins can then associate with additional transfer RNAs ready for the next step in elongation. So if we look at what's happening here in the ribosome in a bit more detail, which is the formation of this peptide bond. So here are the residues we were looking at in the P site and the A site. The peptide bond forms through nucleophilic attack from this amino group here to the carbonyl group here. And then you have the formation of a peptide bond catalyzed by the RNA of the large subunit. And the growing polypeptide is transferred from here from this transfer RNA in the P site to the one that's in the A site. So now the A site containing transfer RNA has the growing polypeptide chain, which is emerging here through the ribosome. So effectively, the growing polypeptide chain is transferred to the transfer RNA that's in the A site, and then the ribosome is able to move forward through to the next codon. So effectively now, the A site is again open for the next transfer RNA to come in, and in the E site, the remaining transfer RNA can now exit. So effectively, we have a movement of the ribosome along the messenger RNA. Now, this is an energy-dependent process, and the energy is provided by hydrolysis of another GTP. And it's thought that the hydrolysis of the GTP causes a three-dimensional change in the ribosome, which allows it then to move along the messenger RNA. So in this animation here, I'm showing the involvement of the elongation factor and the hydrolysis of the GTP to drive the ribosome to move along the messenger RNA. I'm also showing the base pairing between the transfer RNA and the messenger RNA and the exit of the transfer RNA. So there's a lot going on in this animation, but it kind of gives you a hint of the process. So there's the hydrolysis of the GTP, which drives a conformational change in the ribosome so it can move along the messenger RNA. And then you have the next amino acid residue coming in, base pairing, and the peptide bond formation, exit, entry, and the process repeats again and again. So the growing polypeptide is transferred to the incoming amino acid, and then the enzyme activity of the ribosome catalyzes the peptide bond formation, and then the hydrolysis of the GTP drives the ribosome movement along the messenger RNA. So again, some dot point summaries from the textbook. The first step in elongation is an incoming amino acyl transfer RNA binding to the ribosome with the help of this elongation factor TU. This step involves the GTP hydrolysis, which pauses and allows the proofreading of the transfer RNA, and then we have peptide bond formation between the two amino acids that are in the A and P site, or the growing polypeptide and the amino acid in the A site. And I also showed you a diagram which describes the formation of the polypeptide bond through nucleophilic attack of the incoming amino acid to the growing polypeptide chain. And the third step of elongation is the movement of the ribosome along the messenger RNA. So this is that translocation step we were talking about. And this shifts the position of the transfer RNAs from one site to another, and therefore allows the additional transfer RNAs to enter the ribosome and exit the ribosome. So now that we've covered initiation of protein synthesis, we've covered elongation of protein synthesis, let's assume that enough amino acid residues have been added to create the polypeptide. The last stage in translation is the termination of protein synthesis. So the termination of protein synthesis involves the ribosome coming across a codon which is called a stop codon, such as AUG or UAA, as I've listed here. Now, there are release factors which bind to the ribosome once the ribosome reads these codons. So rather than bringing in a transfer RNA into the ribosome, at these codons, a release factor, which is a mimic of a transfer RNA, is brought into the ribosome. So a protein, small protein that mimics a transfer RNA, binds to these codons, and rather than transferring an amino acid onto the growing polypeptide chain, a water molecule is transferred onto the polypeptide chain, which releases the polypeptide from the ribosome. And here's an animation that shows the process of termination. So in this animation, the ribosome has translocated through to a stop codon, and at this stop codon, a releasing factor rather than a transfer RNA will attach to the ribosome. Also shown here is the growing polypeptide chain exiting the ribosome. Excuse me. And the release factor here doesn't bring an amino acid, so when this catalytic activity happens here, a water molecule will be added and then the protein will be released. So there's another protein, RF3, that's involved in this process, and the release factor here resembles a transfer RNA, binds to the stop codon, and then we have the transfer of a water molecule rather than an amino acid to the polypeptide chain, which is then released. We then have the an interaction with the GTP molecule and the release of the release factor 3 here. So there we have the termination of protein synthesis. Again, there's a bit more detail in the textbook showing, describing this process. So again, referring to the dot point summaries in the textbook, after many elongation cycles, we have termination of protein synthesis due to the ribosome reading a stop codon. At the stop codon, release factors recognize the stop codon and terminate peptide synthesis and release the peptide. And during the formation of a polypeptide bond, at least 4 high-energy phosphate bonds are consumed. So we have, the ATP being consumed during amino acid activation, and we have GTP being consumed during translocation and peptide bond formation. And this consumption of energy is thought to improve the fidelity of the process and drive the process forward. So now that we've covered the main points of translation, I'd just like to show you a short video which is a really nice representation of this process. This video was produced at the WeHi Institute, which is in Melbourne. It's the Walter and Eliza Hall. So I'll show this, and I'll probably monologue over the top a little. And if you want to watch the version without me monologuing, then go down to the WeHi's website and have a look at some of the videos they've got there. So as you can see on the screen, here's the nucleus of the cell. This isn't a cell, but this is the nucleus within a cell. Okay, and here's the cytoplasm of the cell. Now, these little yellow structures here are nuclear pores, where proteins can traffic in and out of the nucleus, depending on whether they have a nuclear localization signal or not. I'll cover that in a few slides' time. Now, what's shown here is a messenger RNA that's just been transcribed within the nucleus. So the messenger RNA leaves the nucleus through these nuclear pores into the cytosol, where it will be translated. So here we have the messenger RNA and the bases shown, and here we have the small subunit of the ribosome, which is binding to the messenger RNA. So here you get a great feel for the idea of a molecular machine. So you've got this large ribosome here with the small and large subunits coming together. You've got these transfer RNAs coming in to the A, P, and E site, donating their amino acid that's being joined onto the growing polypeptide chain, and then the transfer RNA is ejected from the ribosome, and you can hear the ribosome clicking along as it moves like a machine across the messenger RNA. And here we have a representation where they've taken away the large ribosome artificially, just to show you an inner view of what's happening. So here's your A, P, and E sites, and you can see the transverse RNAs coming in. There's the peptidal transferase activity happening here, and then the transfer RNA without the amino acid is then departing. And here's the growing polypeptide chain. So here's the codon-anticodon interaction of the incoming transfer RNA. And this is just showing that in the cytosol, there are many transfer RNAs attached to each of the 20 amino acids. So there's a specific transfer RNA for every amino acid. And I guess This is the charged amino acid here, shown in red. And this is just obviously the polypeptide chain leaving the ribosome as a string with no structure, but you can see it starting to fold into an alpha helices as it emerges. So it's starting to adopt a secondary structure and a tertiary structure as it emerges from the ribosome. Lastly, I'll just cover a few points on protein folding, and we'll have a quick look at covalent modifications of proteins and targeting of proteins. So, as translation occurs, the polypeptide exits the ribosome as a string, if you like. Now, as the polypeptide emerges, you start to get the formation of local structure such as alpha helices and beta sheets as the protein's being exiting the ribosome. And then these secondary structures interact to form higher level or higher order structures in the protein. There are also proteins called chaperones which can help the process of protein folding. Many amino acid residues in proteins can be modified after translation. So this is referred to as post-translational modification. For instance, up to half of eukaryotic proteins have a acetylated N-terminal residue. Other post-translational modifications are shown in this diagram here. So for residues that have OHs on them, such as serine, threonine, and tyrosine, the OH can be modified to add a phosphate group. So certain residues on amino acids can be phosphorylated. Also, certain residues can be carboxylated. Certain residues can be methylated, as shown here. So the charges and bulkiness and structure of these amino acid residues can be modified post-translationally through chemical modification. Okay? And these modifications are very important to control protein activity in the cell. Also, proteins need to be located in the right region of the cell to form their function. And there's various methods for targeting proteins to different regions of the cell. One of the best understood methods of targeting proteins involves the ER, which is the endoplasmic reticulum. Many lysosomal and membrane-bound proteins, or proteins that are secreted, are initially synthesized on ribosomes which are actually attached to the endoplasmic reticulum. So here we have some endoplasmic reticulum surrounding the nucleus, and this is referred to as rough ER, as was observed down electron microscopes. Now, these ribosomes synthesize proteins and inject these proteins into the ER, okay? And then the membrane can branch off into smooth ER and other organelles such as Golgi complexes and things. And these proteins can be modified within the environment of these organelles and then can be secreted or located on the membrane through this method of protein trafficking. So these proteins don't go into the cytosol, they're encapsulated in membrane and then translocated through the cell. And this is just showing a zoomed in sort of section of the protein inside these smooth ER in this case. And then these migrate through the cell and can be released or embedded in the plasma membrane. Many proteins contain an N-terminal sequence which plays a role in directing that protein to its final destination. And this is again referred to as protein trafficking. And once the protein has been located, often this N-terminal residue is cleaved away from the protein, is cleaved off. So this is just showing the N-terminal residue here, and this is the sequence that is involved in trafficking the protein. And here is a cleavage site, so later on in that protein's life, this region is cleaved off. And this is just showing some of the examples of proteins containing these sequences, and there are many of them. So while proteins are in the ER for trafficking, they can also be post-translationally modified. For instance, within the ER, some of these signal sequences can be removed. Some of the environment of the ER helps certain proteins fold. Disulfide bond formation occurs in the ER, and also protein glycosylation, so the addition of glucose or sugars to proteins, also occurs in the ER. There's another class of signal sequences that are really important for proteins, and these are nuclear localization signals. Now, many proteins made in the cytosol move into the nucleus. Okay, so for instance, DNA polymerases and histones and all these sorts of proteins that interact with DNA have to be trafficked into the nucleus because they're made in the cytosol. Okay, so these proteins contain a nuclear localization signal, NLS, referred to in the textbook, and these signals direct the movement of these proteins into the nucleus. Now, these sequences aren't cleaved off, and these signals don't always occur N-terminal. They can be anywhere in the protein. And the reason they're not cleaved off is that during cell division, the nucleus dissolves and reforms. So these proteins have to be re-trafficked back into the nucleus during cell division. So these proteins, the nuclear localization signal is a part of the protein sequence that is not cleaved off. There's a little diagram here from the textbook showing a protein that contains a nuclear localization signal in the cytoplasm interacting with some of these helper proteins to be transported through a pore into the nucleus. And then it can then bind and interact and do its function in the nucleus. And just a brief summary of some of the things I've been talking about is that as proteins are made off the ribosome, they start to form 3D structure, such as alpha helices, and then you get cooperative folding of the protein sequence. There are helper proteins involved in this, such as chaperones, and many proteins are further processed by post-translational modification, such as phosphorylation or methylation, and this is important for controlling protein function. So that concludes the lecture covering protein synthesis. I hope that makes sense to you, and I hope the video and animation has been of use to you. Don't forget, refer to the textbook and read through some of the concepts within the textbook just to build up your knowledge base so that you can understand the concepts. Thank you.